All right, you're here with Mr. Price. We are working through Unit 4, which is Photosynthesis and Cellular Respiration. We are going to start very quickly with what is a calorie? And a calorie, if you look down here, is the amount of energy needed to produce 1 raise or 1 degree increase in a thermometer Celsius for 1 gram of water. So if you were to take this marshmallow and burn it and light it on fire, what would happen is the temperature, once this thing lights on fire, will actually increase in Celsius the water over here, and will indicate that on the thermometer. So you can see that it's burning, and we can measure calories in this way. Please understand this is a very crude method, but that burning of that marshmallow and the calories that we get out of a serving comes from that idea that we can raise 1 gram of water 1 degree Celsius by burning our foods. And we do this in a slower rate in a process called cellular respiration. And there's 3 steps of cellular respiration. First one, and always the first one, is glycolysis. The next one, we move into the mitochondria in the presence of oxygen, which makes this an aerobic process. Into the Krebs cycle in the matrix, which is this area right here, if I'm very good, the swimming pool looking area of our mitochondria. And then the next one happens in the cristae, which is the folds here of the mitochondria. And what happens is through a process, these 3 steps for cellular respiration, we take this glucose molecule, this 1 glucose molecule, and what we'll make is a total net gain of 36 ATP. And in the glycolysis, the first step happens in the cytoplasm of cell, and that what happens is we basically take this molecule, add a little ATP, change the molecular structure of it, and turn it into a net gain of 2 ATPs, 2 pyruvic acids, and 2 NADHs. If there is oxygen present, again, we'll continue on this aerobic process and move into the Krebs cycle, which happens again in the matrix of the mitochondria. This is where we get our CO2 made in our chemical formula. You breathe out CO2, it comes from the Krebs cycle or inside the matrix. We get a little ATP, but the biggest thing that we get out of the Krebs cycle are NADHs. Excuse me, my writing's terrible. NADHs. And we also get FADH2 or NAD and FAD. And these guys carry electrons, and they carry electrons to the electron transport chain. And if you were to look right here, and I'll try to draw, yeah, right there, the cristae or the folds, the inner membrane here of the cristae is where that next step of electron transport chain happens. And we'll talk about that here a little bit more in detail. But again, the 3 steps of cellular respiration. Glycolysis in the presence of oxygen. Now we move to a completely aerobic process in the mitochondria into the Krebs cycle, and then the Krebs cycle carries or makes electrons for the electron transport chain, and that's where we get most of our ATP made, and the last byproduct which we breathe out is O2. So things we breathe out, CO2 and water. Now what we need to ask ourselves is this: what happens if there is no oxygen present in us? We get what's called lactic acid fermentation, and so this is a completely anaerobic pathway. So if we go this way, we stay in the cytoplasm of the cell, and we do a completely anaerobic process, and we get this lactic acid and NADH electron carriers to try to make ATP. It's not a very efficient way to make energy. Other creatures do strictly this, like yeast and other microorganisms. They'll do what's called alcohol fermentation. Well, they'll get alcohol. They'll make also CO2, which causes bread to rise, and they'll get some NADH also out of this process. All right, so again, here's the Krebs cycle a little bit more. We are looking at inside the mitochondria in the matrix. We get our pyruvic acid that came from glycolysis. We move into the matrix of the mitochondria, and we basically start adding enzymes. And as we add enzymes, it allows us to make NADHs again, which are going to carry electrons to this, the electron transport chain in the membrane of the cristae. And as we start adding enzymes. We move these carbons around, and as we move them around, we are able to release electrons. They are picked up by NAD. Carbon dioxide is released. Again, another enzyme enters. We move the carbon away. It allows us to get to the electrons. Carbon dioxide is released. Here we get a little ATP. Move the carbon again with the enzyme, and FAD comes in, or FADH2 comes in and picks up electrons. And we keep basically going through this Krebs cycle, moving the carbons around to get electrons moved. And we get all those electrons, and they get dropped off in the cristae membrane, which is this right here, of the mitochondria. So NAD comes in, drops off those highly charged electrons. FAD also comes in, drops off those highly charged electrons. And because it's negative here, and I want to point out this, because we got a negative charge going on in our membrane here, that negative charge draws these hydrogen ions, which are protons, that were stuck out here. So if there's any hydrogen ions out here, they're going to be drawn through inside the inner membrane of the cristae. So all of these, hydrogen ions are brought in because of this negative or electronegative charge that are brought in by NAD and FAD. Why do we want to get a whole bunch of hydrogen ions inside the membrane? Because of that simple principle of high to low concentration. So I have a high concentration here. I'm in the matrix of the mitochondria. I have a low concentration here, and that high concentration naturally wants to go back to a low concentration. So that high concentration goes through what's called ATP synthase. And ATP synthase will take ADP and recharge it into my little battery here of ATP. So I called my battery charger ATP synthase, and that ATP synthase here will spin because of the positive going through the electronegative membrane, and spin the ATP synthase, taking ATP and hooking, or ADP and hooking ATP up to it. Again, this is step number 2 and step number 3 of cellular respiration. And remember, there's 3 steps cellular respiration: glycolysis, Krebs cycle, electron transport chain. This all requires oxygen to happen. There's the oxygen, there's the water that we breathe out, and there's the carbon dioxide that we breathe out. All right, so next, I bring up this slide to remind you that we are heterotrophic organisms, which means we must consume energy. And we consume energy to break it down basically into this. So we'll take glucose. From one glucose molecule, we'll make ATP out of it 36. Remember, ATP represents, and this is what the molecule actually looks like, adenosine or adenine triphosphate, which is a fully charged battery because we got all 3 phosphates there. And then adenosine diphosphate or adenine diphosphate, we have 2 phosphates, which means we are partially charged. So again, we are heterotrophic. We have to ingest foods and other things to make ATP out of that food. Autotrophic organisms, which are producers, take in sunlight in photosynthesis in the light-dependent reaction and convert that light into energy in the form of ATP, and that ATP gets sent to the Calvin cycle to make sugars, which is what we get out of the Calvin cycle. So again, heterotrophic organisms, that's what we are, and other consumers like herbivore, omnivores, those things. And autotrophic organisms are like plants. They produce all the energy in an ecosystem. Here is a slide that deals with the scientists, and we'll start out there. Helmont, I call him the water boy because he figured that most of the mass from plants comes from water. So here's Helmont watering plants. Please note that the plants also gathered in CO2 out of the atmosphere to create the structure that holds in most of the mass of a plant, which is water. Priestley over here showed a gas exchange. Again, we had a mouse, put it in with a candle, mouse ends up dead. We ended up simplifying this even more than what you see here. Priestley put a candle in with a mint leaf and saw that there was an ember glowing with the mint leaf. He showed that there was a gas exchange, meaning the candle gave off CO2 that the plant needed, and the plant gave off oxygen, which the candle needed to continue burning. So I called Priestley the candle boy. And then down here, if you can zoom in on this, you can see little tiny bubbles coming off this Elodia plant. And this is Ingenhouse's bubbles. And what he showed is in the presence of light, oxygen bubbles are made in plants. And he used water plants to show that there are oxygen molecules being formed underneath their leaves. And that's the gas that's released in photosynthesis. What does photosynthesis take in to get these bubbles? It takes in carbon dioxide into the stroma. It takes in water into the thylakoids in the granum, and it produces glucose in the Calvin cycle in the stroma, and it produces oxygen in the thylakoid membranes. Again, this all happens, photosynthesis happens in the chloroplast, and that light energy, the photon energy that comes from the sun, hits that thylakoid membrane to start the light-dependent reaction. The light-dependent reaction will make NADPH, ATP, and send that to the stroma so the Calvin cycle can make glucose. That's why they're called producers, because everything we eat can get tied back to an autotrophic organism. So again, as we get more specific here, the light-dependent reaction depends on light. The Calvin cycle doesn't need light. It's also called the dark cycle or the light-independent reaction. What do we get? We get ATP, we get NADPH, and we take in water in the light-dependent reaction. The oxygen actually comes from the water molecule. So we'll get oxygen out down here. The Calvin cycle will take in the ATP, NADPH. What will it make? Glucose. But it takes that CO2 out of the air to make the sugar molecule. So over here, we have basically kind of the opposite of the Krebs cycle. We're taking the CO2 out of the air and adding energy. And as we add energy, we add the ability to connect carbon dioxide or carbons together. And those carbons get rearranged into what we know as glucose, which is down here. If we move over to the first reaction that happens, which is the light-dependent reaction, we take in a photon of light. That photon of light hits the chlorophyll A molecule, which is the main pigment for photosynthesis. That chlorophyll A molecule gets enough energy to split the water molecule. Oxygen is released, and we get electrons, which are stuck, again, kind of in an electron transport chain in this thylakoid membrane right here. So where are we? We're right here on the thylakoid, and more specifically in the thylakoid membrane. This electron will start losing energy and draw in, because of the negative charge here, draw in hydrogen ions. So we're going to get a high concentration of hydrogen ions in here. And the electrons, again, are following energy. So photosystem 1 comes, we get another photon of light hitting that chlorophyll A molecule, re-exciting that electron, and then our NAD comes in, NADPH backpack comes in, picks up those electrons, and takes them to the Calvin cycle. Now, because we have a higher concentration of hydrogen ions in the inside of the thylakoid membrane, on the inside of the thylakoid in the luma, what will happen is those hydrogen ions will move through an ATP synthase to make ADP into ATP. So again, it's kind of the same principle as the electron transport chain of cellular respiration. We use the electronegativity of the electrons to pull in hydrogen ions. We use the sun to help keep those electrons excited or have more energy potential. NAD takes, with a P, takes the energy to the Calvin cycle. We take the ATP, which gets converted from ADP into ATP from the ATP synthase, and we take that ATP to make sugars. So what are the things that happen in photosynthesis? We take in water and light in the light-dependent reaction to make oxygen. In the Calvin cycle, we take in CO2 and make sugar. So photosynthesis is both of these, the light-dependent reaction plus the Calvin cycle making photosynthesis.